Thanks, Carla. So welcome everyone to the invited session on genome mapping and sequencing technologies for characterizing structural variants. So I'm Rashmi Kanagal-Shamana from MD Anderson, and my co-moderator is Bryn Levy from Columbia Medical Center. And we are all honored to have two distinguished speakers today, Dr. Lisa Lansdon and Dr. Fritz Sedlazeck. Just one point is, as with any other session, the Q&A will take place after both the talks. So please hold on to your questions until both the talks are done. And when it's ready for the Q&A, please line up behind the two microphones on either side to ask your questions. Bryn?
Okay, it is my pleasure to introduce Dr. Lisa Lansdon, who is an Assistant Director of the Molecular Genetics Lab in the Department of Pathology and Laboratory Medicine at Children's Mercy's Kansas City, as well as an Assistant Professor of Pathology at the University of Missouri, Kansas City School of Medicine. Lisa is the chair of the CGC Macrogenomics Event Detection in Solid Tumors Working Group. She serves on CGC's Education Committee and AMP's Membership Affairs Committee, and is a technical editor for ACMG's Genetics in Medicine journal. Her current research interests include exploring the clinical utility of long-read sequencing and RNA expression profiling in pediatric cancer diagnostics, as well as assessing the strengths and limitations of existing and emerging technologies utilized to detect macrogenomic events in solid tumors. So welcome, Lisa.
Thank you, Bryn, for that very kind introduction. And thank you very much to the program committee for the opportunity to share some of the exciting new developments that we're having at Children's Mercy with you all today. So here, I will openly profess that the somatic structural variant detection, does a Goldilocks solution exist? I cannot promise to share with you a Goldilocks solution today, but hopefully my goal is to be able to bring a few different ideas to the table about what different types of technologies are currently available, maybe those that are emerging, and what you might be able to employ clinically, if not now, in the near future. So as was mentioned, my disclosure is that I am on the editorial board for Genetics in Medicine and Genetics in Medicine Open, and I do receive a small stipend as a result of that work. So for those of you who appreciate a roadmap, here you go. We have two parts to the talk today. I will warn as well that it's going to be a marathon of data in a sprint session, so please buckle up and bear with me. For part one of the talk, we are going to be going over the technologies that currently exist for structural variant detection, and also taking a step back and discussing what are structural variants, what's categorized for the purposes of this talk. Then we're going to look at newer technologies as well. It will be a very rapid-fire brief overview, but happy to discuss any of that in the Q&A or later today or tomorrow at the meeting. Then in terms of investigating clinical utility of long-read sequencing, that will be part two. So we'll be talking about what are the advantages, limitations of using long-read sequencing, and why we're excited for that as a clinical tool. So without further ado, structural variation. So these are going to be regions of the genome that are a little bit larger than what we typically think about with sequencing technologies. So deletions, duplications, historically defined as about 1 kb or larger, though that is becoming a matter of debate, I think, in recent years as we've had improved resolution for these types of events. In cancer, the sky's the limit on the complexity, and so it really does get complicated pretty quickly, where we can have these inversions, translocations, multiple events all within the same tumor cell. And so really the goal is what technologies can we harness in the best possible way, the easiest possible way, to try to uncover some of this complexity overall. Why are these important? We have a number of different fantastic talks that have already been given talking about structural variation in cancer in general. At Children's Mercy Kansas City, we're a pediatric hospital, and so part of the flavor of my talk today is going to be very pediatric focused as a result. So here you're seeing the WHO Blue Books Online, a couple of screenshots from some of the table of contents, looking at acute myeloid leukemia and B-cell acute lymphoblastic leukemia. So in both of these, hopefully you can appreciate that the majority of the genetic contributors or genetic defining attributes to these tumor types are going to be the resulting fusion. And so how do we get to the fusion? Well, you need to have some sort of structural alteration underlying the DNA that can result in the fusion that can be detected by DNA, RNA, et cetera. This is not unique to leukemia, though, and so although it was harder to capture on a slide, I do have a shout out for solid tumors as well, and this is looking at other rearrangements that are important, say in sarcoma testing, for example, but there are many, many examples of why this would be very relevant in terms of cancer, in terms of diagnosis, prognosis, and therapeutics. Now looking at technologies that exist. So this is that overview that I wanted to share with you all about we currently implement in the clinic. So here we're looking at chromosome analysis. We have fish analysis, microarray, and then sequencing. This would be short read sequencing for the purposes of this talk, primarily exome and genome sequencing. Goal here is to tell you some of the strengths and limitations, with the caveat being that this is my perspective on the technologies, and I'm more than happy to debate any of these with anyone as the meeting progresses. So here with the strengths, one of the really cool things about chromosomes and also fish, as you'll see when I click my button, is that it's quantifiable at the single cell level. And so chromosomes, as we know, really effective for some of these large-scale alterations, and it also provides this larger genomic context. So we're able to get a sense of clonality, whether something is a primary genetic driver found in all cells in a particular tumor versus subclonal, for example. Fish is very similar in that way, where we can look with single-cell resolution to be able to determine whether or not a structural alteration is present. It can be run on FFPE or fresh sample, which is fantastic for that particular type of modality. It's also scalable. So if you want to look for multiple alterations, you can build out a large fish panel. If you want to look at only a few, you can do that as well. One of the other things that's great is that it's very effective at detecting presence versus absence of a specific structural variant. And you could easily rule in or rule out whether that SV is present. It's also fast, so this could be about a 24-hour turnaround time if needed. With microarray, so what we're looking at for microarray is it's going to be a genome-wide interrogation overall, and so this is more bulk analysis of the DNA. There's FFPE or fresh samples, again, broad strokes here. Also includes copy-neutral loss of heterozygosity, which can be important for certain tumor types. Then we can include additional genetic information for some of these array types, so hotspot mutations, for example, and it's a higher resolution than our chromosome analysis overall, if we're saying that's our baseline, what's been historically utilized for these tumor types. Last but not least, short read sequencing. We're almost done with this slide. With the short read sequencing, genome-wide interrogation, FFPE or fresh tumor, includes CNLOH assessment as well. And then we can also find additional genetic information, so find our SNVs, for example, also pick up smaller alterations. And because we can pick up the smaller alterations because we have the sequence data, this would be things that would allow us to have a little bit higher resolution overall. Then finally, which I think is most exciting about short read sequencing, is we can do it in a paired tumor normal way. Arguably, you can do that. You could fish for a normal sample, fish for a tumor sample. You're kind of doing that within your fish assay anyway, knowing there's some normal cells sneaking in there. But this truly can be a paired tumor normal, subtract out all germline variation, and only be left with the somatic if you desire. Challenges. So here, challenges for chromosomes, very sample specific. So you need to be able to culture your sample to see anything. Also relatively low resolution, which we know. Fish, it's very targeted, very specific. It's a strength and a weakness of this assay. If you don't look for it, you won't find it. Sometimes it's really difficult to resolve these complex patterns that we weren't expecting in the data. Microarray, it's a bulk technique. Don't have that single cell resolution, cannot find balanced rearrangements, difficult to resolve some of the breakpoints depending on where the probe coverage is in your microarray build. Also variability on the complexity. And finally, it's traditionally run as tumor only. Last but not least, back to our short read sequencing. So it's going to be bulk interrogation as well, like microarray. And then there's going to be some variability there. It depends on the platform that you're using, the overall design, what can you actually capture, and we know it struggles with things like pseudogenes or repetitive regions. So now we have the newer technologies. I am incorporating OGM into newer technologies. I think it's newer to the majority of us in the room. And as we know, it's being successfully, actively utilized clinically, which is really exciting. Also Hi-C, so this is going to be a kind of, again, umbrella term that I'm using for a lot of different technologies that are coming out in the Hi-C space. Not yet implemented clinically to my knowledge, though every day it seems like there's a new technology that's being implemented in the clinic. So I'm happy to be corrected here if someone is using it clinically. Then long-read sequencing, where we're going to be focusing a lot of the time today. Not yet implemented clinically, with a big asterisk in that I'm saying in the somatic space, specifically in the clinic. We have successfully launched it clinically for constitutional, which I'll touch on at Children's Mercy, but for the sake of this talk, it's more focused on somatic structural variation. So quick highlight over the 3 technologies I just mentioned. So one, optical genome mapping. For those of you less familiar with this technology, although this is being used in this particular diagram as for prenatal testing, we can swap out amniocentesis or CVS with tumor or your heme malignancy of choice. You isolate ultra-long molecules of DNA. You label them that we're looking for 6 nucleotide repeats that are going to fluoresce when we run them through a particular machine. And what we're able to do is take single-stranded molecules of ultra-long DNA, detect where those different fluorescences have bound, and then we're able to reconstruct the genome and make these calls depending on where those repetitive sequences are. What works beautifully for this is going to be fresh samples or samples that have been fresh frozen with some stabilizer added. And there is a special extraction protocol, so just something to be aware of when utilizing this technology. You need specific instrumentation, you need specialized data visualization software, and then also the resolution, it's going to really be dependent upon where those different markers fall. Then if we're looking at Hi-C, so this is going to be looking at cross-linking the DNA, trying to capture the 3D conformation of how the DNA molecule is at a given point in time. And so the beauty of this is that you can cross-link the DNA, you can then capture where you have those cross-links, and then end up doing an analysis of those rearrangements in a specific way. You can use fresh frozen sample. These should be viably preserved cells, though. Also can use FFPE for some technologies. Again, there's a lot of different groups out there actively investigating this. And there is a specialized DNA extraction protocol, so things to be aware of if thinking about clinical considerations. There's also specific library prep, and you can pair it with this paired-end short-read next-generation sequencing. And so the beauty of doing that is then suddenly you can get breakpoint resolution sometimes, depending on how this is implemented, down to the single nucleotide. If not, it'd be a little bit larger scale if you don't pair it with that follow-up sequencing. Last but not least, where we'll spend the rest of the time today, is going to be long-read sequencing for structural variant detection. So here you can see that we're taking long-read sequence data, and then we're working to align it, and then call different structural variants in different ways. I'm going to be focusing on one primary structural variant caller for the talk, and also explain to you why I'm particularly excited about that caller. And one of the things to know about samples, so we're still talking fresh frozen for now. Also specialized DNA ISO protocol, but that has the caveat of we have found that in our hands we can use our standard run-of-the-mill DNA isolation procedures, and it seems to be working pretty well on long-read sequencing. You also have a specific library prep. You would need long-read sequencing capabilities, which I know is not available at every institution, and the ability to do the SV pipelining. Last but not least, you can do breakpoint resolution down to the nearest nucleotide, which is very exciting for some future considerations. So part 2, here we go. This is looking at clinical utility of long-read sequencing specifically. So why am I up here talking to you about this? Well, it's because at Children's Mercy, we've made a really big investment into genomic medicine in general over the last decade, and I should say 2 decades. We are a very small institution, despite a lot of the progress that we've made in the sequencing arena, where we have about 8,000 employees, and we have approximately a little shy of 400 hospital beds for our pediatric patients that we serve in the Kansas City metro area and also the surrounding region. So our Center for Pediatric Genomic Medicine. It was established in 2011, and coincidentally, we just celebrated last week our 10-year anniversary of offering clinical genomes, which is quite remarkable. We are the first to do it and continue to do so today. There's been a big capital investment in our Center for Genomic Medicine as well, and so this is not just specific to the Center for Genomic Medicine, but what Children's Mercy has done is they've built a research institute, pictured here, that opened in 2021, where our laboratory has moved to and is currently housed alongside researchers, all with the goal of trying to help patients find answers that don't have answers yet. So one of the biggest projects that has been very successful at Children's Mercy is this study that you may or may not heard of. It's in the constitutional space. It's called Genomic Answers for Kids, or GA4K. And in GA4K, this is a collaboration between PacBio and Dr. Tomi Pastanen, who is in the lower left-hand corner of the slide. And what they attempted to do with this skilled team of clinical laboratorians and lab directors is they looked to see whether or not they could find answers for children with rare disease that weren't able to have answers when they went through the clinical pipeline within our laboratory. And so what they've done to date is they've sequenced over 1,500 samples of these patients as well as their parents, looking for answers for these children. Other than to tell you that we have a really well-established long-read sequencing program at Children's Mercy as a result of that particular study, I won't be commenting more on GA4K today. However, if you're interested in the study, it was published, the first approximately 1,000 cases, and those results was published in 2022, and I'd encourage you to go give that article a read. And then as a result of those research efforts, they since have launched hi-fi sequencing, long-read sequencing. We'll talk about kind of the different flavors of long-read sequencing in just a minute. But they launched that in October of 2023, and so that's actively being used clinically on the constitutional side at Children's Mercy today. So looking at these technologies, there's two primary types of long-read sequencing when we're talking about it as a technology. At Children's Mercy, we're actively utilizing hi-fi sequencing, or this would be from PacBio. There's also Oxford nanopore sequencing, or ONT. Both of the chemistries are really interesting in that what they do is they're able to take larger fragments of DNA and with very high accuracy detect the sequence variants that are present and end up sequencing the DNA molecules with really high accuracy overall. So the way that the technologies actually function is that when you have your hi-fi sequencing, there's adapters that are ligated onto the end of your DNA sequence. Then through a series of chemical reactions, and the sequencing reaction is that you end up with circularized DNA that's then sequenced over and over and over to give you a really high amount of accuracy for that particular molecule. Oxford nanopore is really cool from a chemistry perspective as well, where what you actually have are these nanopores. And so you have the DNA that can migrate through each of these little pores during the sequencing reaction. And what happens is that there's actually going to be a difference in the electrical current that occurs. And so with each nucleotide, they each carry a slightly different charge, and those charges then are translated into sequence data. One of the tools that I'm really excited to tell you about today is Severus. Severus is a brand new, hot off the press, structural variant caller for cancer specifically, and it can be used in a paired tumor normal fashion. What I wish I could do is a deep dive into Severus today, and unfortunately I cannot, but fortunately we do have the creators of Severus here with us at the meeting today. And so here you can see Dr. Misha Komolgorov and also his very talented postdoc, Aisha Keskus from the NCI. And if you're interested in Severus as a tool, would highly encourage you to go visit poster 71 during the poster session, and they can tell you all about how they built this tool and how useful it can be. So what we've done is we've taken Severus and we have harnessed it in first a research setting. And we said if we do a small cohort study of 17 pediatric patients with leukemias, some with known alterations, some without known alterations, this is looking at primary drivers that are structural variants specifically, how does Severus do? Can it call them? Can it not? If we have some knowns, if we have some unknowns in terms of overall diagnoses, what happens? So here I am going to gloss over the demographics of this particular study, but I hope what you can glean from it is that we tried our best to have a fair amount of diversity in terms of types of calls, in terms of B-ALL versus AMLs, and what we really wanted to do is put Severus to the test. What you'll see in the lower portion here, and I'll turn on, yes, perfect, is that before we did our sequencing, all of these cases, they ended up going through routine clinical testing. And so pre-sequencing with our B-ALL cohort, we had 2 of them that had known structural alterations that were primary drivers. And what you can see is that we have an increase in colors if we go over to the right. So we did find that there were additional drivers that were identified using Severus as a tool in our long-read-seq data. Similarly, in our small AML cohort, we had 1 that was unknown prior to long-read sequencing and analysis with Severus. All of them were resolved successfully when we utilized this structure and this tool. So how did we do it? Well, we took DNA. This requires a higher DNA input than some of your other sequencing technologies. We're talking about 5 to 7 nanograms for at least the hi-fi sequencing we're doing at Children's Mercy. They were isolated through a variety of different isolation types, nothing super special about the isolation, and this would have been blood and bone marrows. Then we performed hi-fi sequencing on a Revio. This was a single SMRT cell, so it was about 30x coverage overall. Then we did our PacBio WGS workflow, followed by structural variant calling by Severus. So on this slide, I'm going to walk you through one case. This is an AML case where we had done our routine workup, had come up empty-handed, said, maybe we can find something with this newer approach with long-read sequencing, and we did. And so in this particular case, what we're looking at are long-read sequence data that are zoomed into a region on chromosome 10p. And what we found is that this region in chromosome 10p was actually inserted into another region on 11q, and there are some players that this crowd would be very familiar with. You can see that there's a breakpoint in MLLT10, and then if you can see where the breakpoint is within 11q, it'd be KMT2A. So if we're looking at the data a little bit more closely, what you'll see here are going to be reads very akin to short read sequencing, where each of these lines are going to correspond with an individual read. And when you're walking through, say, MLLT10, what you'll notice in some of the reads is that there's going to be this series of what appear to be mismatches in the rainbow colors. And then if you're walking in the other direction, there's going to be a second series of mismatches. Well, when we go and we're able to look at where are these mismatches in the genome, what we see very, very clearly is that there's actually going to be mismatches that correspond with this gene ARMC3 that's further centromeric within chromosome 10p. We're also going to be seeing reads that are corresponding with a portion of KMT2A. And so what this tells us, and what I tried to diagram, hopefully successfully in this upper portion, is that we do have inserted sequence into KMT2A from MLLT10. What does this look like when we are thinking about it from a FISH probe perspective? Because we did FISH for KMT2A and came up empty-handed for this case. Well, here if we're looking at 11q, centromere is going to be on the left, telomere is going to be on the right. We have our KMT2A probe that flanks KMT2A and also extends a little bit into the KMT2A gene. Well, what we found is that with this inserted sequence, that there was a separation of the probes that's about 1.3 megabases. Well, 1.3 megabases is under the resolution of FISH, and so we couldn't see this separation when we looked. If we would go into the sequencing data, what we can recreate, and what's really exciting from my perspective, is we can determine the exact breakpoints. We can figure out exactly where in the intron do these breaks reside, where is the inserted sequence, how did it come about. If we're taking a step back and saying, wait, why was this missed? So I already mentioned to you that FISH had been performed. The insertion wasn't picked up by FISH. And so we know that that was negative. Well, it was cryptic on karyotype. We couldn't see an insertion this small on karyotype. It was balanced on microarray. We couldn't see it on microarray. We only do SNV calling by short read exome. We did go back and look, and because the breakpoints are intronic, it's very difficult to have any support for this particular insertion from our short read sequencing data. And then unfortunately, as happens in some of our cases, RNA wasn't available for additional follow-up. When we're looking at the FISH probes, if you don't believe me that it was an insertion, maybe those of you who are cytogenetically trained will believe me now. We did follow up with the MLT10 probe, and what we did see was clear separation of this break-apart probe, telling us that we do have additional support that this is a real finding, not just something that we stumbled upon falsely within our long-read sequence data. So here, to summarize this small cohort study, what we found is that we were able to successfully identify known and also identify unknowns, or define, I should say, unknown drivers within these cases. So in the final portion of my talk, what I'm going to talk about is where we're taking this next. So we're actively validating this for clinical use. We're incorporating DNA isolated from tumors as well, which is where we now have tissue that we're using. And this is going to be a summary, high level of our validation cohort. So what we're looking at are, we can either do this in a tumor-only fashion or a paired tumor-normal. We're really going to focus only on our tumor-only results for the sake of time today. And so as you can see, we had 35 patients that are in this validation study, and what's really nice about this analysis, with the big caveat being that you have to have access to the long-read sequence data, is once you pipeline it through Severus, it's an Excel macro analysis looking for these primary drivers, and then we can follow up with visualization and IGV. When we looked at these data, what we found is that in 28 of our 31 cases that had clinically known structural variants, we could detect them successfully, and 4 of our 4 negative controls were negative for any clinically relevant structural variants. So what happened with those 3? This, I apologize, it's a lot of colors, a lot of moving parts, but wanted to give you a sense of the cohort that we looked at. It was more heme than it was solid tumor. We had a variety of different rearrangements very purposefully. And then when we zoom in on the 3 that we weren't able to detect, some of you might say, wait a minute, Ewing sarcoma, that's a solid tumor. Well, what we found is this was a patient who had a metastasis, so we actually tested bone marrow, which is why we put it in the heme category for now, but I think we could argue about semantics there. Then we also had a P2RY8 that we weren't able to detect, as well as a FUS-ERG. When we looked at the details, it turns out this all came down to tumor percentage requirements. So even though we were aiming for high tumor percentage overall, we went back and looked at orthogonal data, and we found that all of these were around the 20% or less tumor range. And so with a single SMRT cell at 30x coverage, some of these are just below our limit of detection overall. In addition, we did an LOD assessment. So this was by taking normal reads that were from the paired normal sample and doing an in silico dilution of that tumor sample. We also did a physical dilution where we actually took the tumor DNA, and then we ended up using the normal DNA from that sample and diluted it out to determine limit of detection. And both give us about 20% limit of detection from a SV-VAF standpoint. So we're talking about approximately 40% tumor is what would be necessary for this in our hands with a single SMRT cell. Advantages and challenges. So here, looking at the advantages, can be relatively quick if implemented in specific ways, especially the analysis, very quick in Excel. Genome-wide data generation, you can have accurate resolution of the breakpoints down to a single nucleotide, which is a really exciting advancement if you consider maybe implementing minimal residual disease testing in the future. You can also run it as tumor normal, and we don't really need any sort of specialized isolation protocol. Many challenges yet to overcome. How do you balance depth of coverage versus overall cost? An important question. You need a lot of upfront capital to actually have this up and running within your laboratory, and bioinformatics support is critical. You can only use fresh, frozen. FFP technology and is coming in the long-read sequencing space, but that's still more on a research basis for now. Relatively high DNA requirements, so it won't work for everything. Tumor percentage matters just like other technologies. Is there such a thing as too much data? There might be. We need to store it somewhere. We need to know how to dig through it. Also, it's bulk DNA analysis as well, and then incorporating it into existing workflows. So I think with that, I have one more slide that I'd like to cover, and because I think we're coming up on time. Is that correct? All right. All right, so parting thoughts. Here we're looking at overall, what can we do within the clinic? I am not aware of a Goldilocks solution that exists yet. I think it's lab specific. I think that it depends on the turnaround time that you need. It depends on if you need single cell resolution versus bulk resolution. And so overall, I think that these are important conversations that we can continue to have in this field. I also didn't touch on any of the other variant types that can be detected with long-read sequencing, which is a very important consideration if you're thinking about implementing this clinically. And last slide, thank you for letting me go a little bit over, is many acknowledgements. Cannot acknowledge everyone here in the time allotted, but huge thanks to Drs. Kamologorov and Kaskus for all of their work on Severus. Also huge thank you to Dr. Midhat Faruqi, our Director of Molecular Oncology, and then Biangil Yu, who is our bioinformatician that got all of this up and running for us. And last but not least, huge thanks to the patients and their families for making this possible.
We'll move on to our second speaker for the session, Dr. Fritz Sedlazeck. He serves as an associate professor at Baylor College of Medicine's Human Genome Sequencing Center and also as an adjunct associate professor at the Rice University. His research group focuses on mechanisms of formation of structure variants, impact on phenotypes, and developing computational methods to evaluate them. Dr. Sedlazeck has served and led major projects in multiple national initiatives like TopMed, CCDG, CAR dementia projects, as well as all of us, advancing both short-read and long-read sequencing efforts. Please join me in welcoming Dr. Fritz.
Yeah, thank you so much for inviting me. It's really an honor and a pleasure to be here and talk to you about our newest developments. Also, thanks to Lisa for amazing introduction. I would have skipped a bunch in my talk unintentionally, so thanks for picking that up. So for disclosure, I got some research support from Illumina, Nanopore, and BugBio, and also the second half of the talk is about a project where we collaborated with Genentech. As Lisa already introduced us, structure variants is really a phenomenal and very interesting genomic alterations of usually defined as 50 base pairs and larger, being described often in 5 different types of structure variants, being deletions, insertions, duplications, inversions, and translocations, or combinations of them that we are learning more and more over the recent years. The interesting fact is they usually cause a higher diversity than single nucleotides on their own, despite being just 0.1% in terms of numbers of SNVs. Over the last couple of years, my group and many others have highlighted the advantage of long-read sequencing, and that's also part of why I'm so excited about this meeting today. We're talking about them, but also about structure variants and the importance of structure variants in cancer studies. So when it comes to structure variants, long reads are still much better in detecting these type of events, which we usually can, in a healthy individual, is around 25,000 to 30,000. To the point of Lisa, is there ever too much data? Probably there is. With the short reads, we typically detect 10,000 to 12,000. One of the reasons of that is because of insertions, such as Lisa just showed in her previous talk. But the other reason of that is that around 70% of these structure variants are occurring in tandem repeats, so in some kind of repeats, which is really cumbersome to kind of characterize correctly with short reads. And over the years, there have been multiple catalogs published. In 2022. We published one in Nature Biotech about 400 medical relevant genes, even in healthy individuals that are hard to assess with short read data. But really then the collaborations of T2T, where we fully resolved the whole complete human genome over the last 2 or 3 years, published already, really drove home the message of that we can now get into centromeres and telomeres, where often also structure variants are happening. So needless to introduce you guys to why we are caring about somatic mutations in cancer in general. But of course, there are multiple driver mutations. Some of the mutations might be not driver mutations that we are looking for. And typically, or very often, it's comparing a normal versus a tumor sample that have been sequenced together. Over the last couple of years, we have developed SNIFLES-1 and then SNIFLES-2, which was published last year in Nature Biotech early on. And so in SNIFLES-2, we took a more general approach of combining and comparing variants. So like what SNIFLES-2 can do is it actually computes a little binary file, which is around 20 MB of size. You can actually share that with collaborators on the per single sample level. And then we can merge and compare do kind of a joint calling approach across 2 samples, or 4 samples, or multiple samples. So in one go, you can get the whole tumor normal pairing, or you can get even metastasis in there, or even other samples to compare to. Currently, our record is around 20,000 samples merged together in this kind of fashion and analyzed. So over the years, we have been also interested in cancer genomics. That's hopefully the reason I'm here and speaking to you all. So here's just one example, which is now very, very much straightforward, either with Severus, as Lisa just described, or with sniffles as well, where we just highlight one of the mutations, a P10 deletion, which deletes 2 exons in COL829. COL829 has been really becoming kind of a nice benchmark point, but also a pain point. And we most recently have put out a publication in Genome Research explaining this a little bit more. So in our study that we just, as I said, published in Genome Research, we characterized all different samples that have been published around COL829 over the years. So there has been studies back then in 2010, so there have been multiple attempts to catalog and structure variants. A multi-platform study, for example, from 2022, more recently, has cataloged around 70 structure variants. When we look at IGV, and I'm kind of apologizing, these IGV screenshots will come a lot in my talks, but I guide you through. So like each of the lines down here is a read, similar to what Lisa just showed you, and up here is the coverage track in gray. So you see that we have an increase in coverage in all different 4 samples. So here's Nanopropag Bio that we sequenced, and then there's 2 older samples that we obtained from different sequencing centers. And so you see that while this is COL829, there has been some remarkable similarities in the coverage, right? We see all these coverage increase around this spot here, going to this spot. But the interesting thing is, for example, in this sample here, there's no inversion present that is indicated by these colorful reads here, right? And so this made us really wonder how reliable and how robust this, first of all, 70 somatic SV catalog is, but then in general, if we have to rethink COL829 as a benchmark sample. And indeed, when we went into this, as I said, with characterizing multiple resources, we only found around 50 SVs in this COL829 that are actually stable. And these were published together with all the results in Genome Research. Another thing that we wanted to assess also is, like, of course, the reveal of the T2T genome, or the CHM13 T2T genome. And this brings multiple benefits, as we could show in the science articles that we have published together with an amazing team of collaborators. So first of all, it improves the mapping of short reads as well as long reads. This is due to the fact that in GRCH38, we have lots of regions that are not well characterized, such as ends, for example, or wrong repeats, or even wrong representation of genes. We published an earlier article highlighting 33 coding protein genes that are wrongly represented in GRCH38. So like, there are many benefits of utilizing CHM13 as a reference genome for your analysis. And we could show that it really improves the somatic SV detection, basically lowers the number of false positives in this comparison between tumor and normal. There are different challenges, of course, and one of the biggest ones you probably wonder is like, how well is it annotated, right? And the frank answer is like, well, not as great as GRCH38. We have many more annotation resources and knowledge about 38 and all the genes that are involved. And this is really something that we try to overcome. So we were wondering how we can compare this and how we can improve this utilization maybe of T2T for the future. Oops. So here's one example. Again, apologies for the IGV screenshots. But basically, look at the bars down here a little bit. So this is one SV where the blue bars down here represent the structure variants in GSH38 on the top, and then on T2T in the bottom. And the interesting thing is, like, although they are same type and same location, they are different sizes, which just comes as a surprise when you use different reference genomes, right? It kind of makes sense that not everything in the reference genome is the same between T2T and GSH38, just different underlying samples. So we really started to wonder, well, how can we compare these SVs across references and then utilize the annotation resources of GSH38? Because just the liftover, as we are sometimes used to with SNVs or point mutations, it's not going to work because the structure variants are in repeats. The repeats are sometimes differently located or differently represented. So it's really hard. And so the thing that we developed here is, instead of using coordinates or SV types, we can actually go back and use the reads themselves. So Sniffles, for example, reports the read names for each mutation that it supports. And we can use the set of read names in the VCF files to compare the SVs and assign them to each other. And this is really something astonishing because basically using this method, we actually found flips of SV types. So what is an insertion maybe on 38 might be a deletion on T2T, and also flips in different locations, even across chromosomes, on the two reference genomes. And this comes down to the representation of different tandem repeats in these two different reference genomes, which was quite interesting and remarkable. So that's why I wanted to highlight this to you guys today. Obviously, structure variance is not everything, right? As well as SNVs are not everything. So like we have been thinking a lot over the last couple of years how to leverage different variant resources and different epigenetic status all together to bring it together in one comprehensive overview of the sample or the tumor sample that you have at hand, or the patient for right? And so we developed multiple new technology, multiple new methods, and this is a development that we did in collaboration with Genentech, Christian Hammer. So we developed an LOH caller and a CNV caller, which is called Spectra. Sniffles, I just talked a little bit about it, and it was published last year. And then we also tried to incorporate methylation. So again, to the point that Lisa brought up, that, sorry, I'm quoting you all the time, but like, can there ever be too much data? Yes, definitely, but we are trying to help you guys in this. And so we put together an entire framework with these new tools to basically, first of all, was just the goal to assess hard and soft loss from a long-read sequencing perspective, but then really extending this genome-wide. So this framework automatically goes in, calls SNVs, SVs, and CNVs, so structure variants and copy number variants, using the SNVs for phasing and giving us additional powers. And then on the CNV calling, we can also do LOH assessment and then phasing. Now, what is important to realize, I think, is that many of these tools are available for short-read sequencing, right? So like, when I tell someone, oh, we can now do LOH or copy number calling on long reads, and one's like, yeah, well, we can do that since 20 years on short reads, right? So what's the fuss about, right? But it's actually really cool to come to a similar level maybe than short reads, because copy number calling, for example, is much more tricky in long read sequencing, because actually the number of reads that we have that are used for copy number calling is much reduced compared to short reads. So it's not so easy, quite frankly. So using resources from Genentech and sample sequenced, and we did, of course, a couple of studies. We sequenced multiple cancers, as you will see in the next couple of slides. So, for example, here is a pretty standard, straightforward tumor normal from ovarian cancer cell lines, where we see the copy number in the tumor, and we don't see the copy number in control here in blue. And so that's cool and easy. And we see also here LOH happening in the tumor, where in red we have the same SNPs marked than in the normal. And here the blue SNPs are basically gone in the normal that indicate the heterozygous variants here. There's still some noise because it's long reads, not everything is perfect. But overall, this is kind of straightforward. And we can assess HLA and other things with that. We can also do it genome-wide. So here's just one visualization from our postdoc Louis about LOH. And variant allele fraction, so red is 100%, and you see we have a lot of LOH going on in this, on a couple of these chromosomes in this particular tumor sample. But of course, this is kind of straightforward, and maybe that's already what you have heard about multiple times, but like, you guys should come back to me and say like, well, Fritz, but this is never as clean as you're just presenting it, right? So one of the big struggles we had over the last year or two was tumor contamination or tumor purity, right? Which is hard to deal with, quite frankly. But we took up the challenge and incorporated this, which we had to, quite frankly, in the different stages of this pipeline. So it can now take into account tumor purity. So to show you that this works a little bit at least, you have to take my word for it, of course, is like here's again a pure tumor with a control. Same scheme, so the red dots are the ones that are indicating LOH in our opinions, and then the red dots are marked in the control just for tracking, and the blue dots are basically absent in the tumor. And so we mix these two samples together to kind of just simulate our boundaries and see how well it goes. So in 90, 70, and 50% of tumor purity, we can still assess LOH quite nicely, and the red dots are just tracking from the 100% tumor purity, how they behave across the different purity ranges, right? And quite frankly, after 50%, it gets a little bit shaky in our pipeline. It gets a little bit tricky to still identify LOH. Some other things like structure variants and so on are still possible, but LOH and methylation gets a little bit tricky, quite frankly. So we performed this analysis across 13 matched tissue samples, 9 sarcomas, cell sarcomas, 2 large cell sarcomas, and so on and so forth. The tumor content reached from 50% to around 90%, and coverage-wise, we basically sequenced each sample with 1 flow cell of oxide nanopore. Here's just a table with the entire overview of SVs and other variants that we called. So in this kind of analysis, what does long reads give us besides structure variants? And I think this slide might show this very nicely. So on the top, you have the normal. You have 1 haplotype here, a little bit grayed out, but this is kind of haplotype 1, and this is haplotype 2, where you see there are more structure variants in terms of insertions and deletions going on. So it's quite an interesting one. And below, I'm showing you the tumor content, right? So the tumor has a 70% purity. And so what you can see nicely now, because we use the phasing, for phasing the tumor, we use the information from the normal, is you can see down here the reads are actually not the second, are actually the second haplotype from the normal cells, right? So we can really separate these things out really nicely. And then on top of this whole thing, you see these red tag marks here in the green box. That's where we can see an increased methylation that basically hypomethylates this exon in the tumor itself compared to the normal. There's also a little bit of methylation going on around here, but this is much more clearer, right? And so these long reads really can then give you all these resolutions in terms of SNVs, SVs, CNVs, and methylation in one assay, in one go. Yes, it's more expensive than optical mapping or other assays or Illumina, but it really can give you this comprehensive overview if you have the right tooling and the right support. And so with the same sample, we did this also on genome-wide, right? So here, for example, I'll show you plots per chromosome. And it's just focused on methylation, so that's the lung 5 sample, 70% contamination. It's purity, sorry, I always confuse these two. And so you see the methylation status of the tumor, the methylation status of control, and this is just one of the plots that Tumor Lens produces for you, together with like tons of tables in terms of there's too much data. But in black, you see where the tumor and the normal have equal methylation status, and in blue, you see all these regions in the tumor that have lost methylation. And then when you squint your eyes, and hopefully you believe me, there are a couple of red dots here and here and here. These are regions or genes where the tumor has increased the methylation. So basically epigenetic knockout, if you want to see it like that, potential. And so this is, I think, very powerful, right? Because like, yes, we can finally call LOH on tumor samples, which Illumina has done for 20 years almost. But we can also put this together with methylation and other variants in a comprehensive way. And then just to make the point, we can also call copy numbers. So this is the same sample looking at the copy number profile. In red, duplications, and in green, deletions, so that you can see we can not just do smaller structure variants, but also kind of chromosome arm alterations and other things, all in the same pipeline put together with nice outputs and visualizations. So I think I'm rushing it, or I'm a little bit early, but kind of the summary of this tool, which we call Tumor Lens, I hope it's a good name and rememberable, is really to bring together all these different variant types, SNV, CNV, sex structure variants, LOH, and methylation together, right? And the reason for it is because they co-occur together. Like, it's not just that there's a tumor that is only based on SNVs, or there's a tumor only based on structure variants, right? So we need to kind of mature up and look at all these other tricky mutation points at once. It can assess genome-wide methylation, as I just showed you. Here's a reminder of this plot where we really see nicely hypomethylation and hypomethylation, hypero and hypomethylation. And you get a nice table and a nice visualization for that, as well as really a special focus, and I didn't have time for that, to go into HLA, use HLA as typing to infer which kind of haplotypes the patient has, and then remap and reanalyze the reads automatically based on the typing results, independent of tumor purity. And then, as I said, like up to 50% purity we can still handle. Below 50%, it gets a little bit tricky, quite frankly, with LOH, for example, methylation. CNVs often gets a little bit lower. SVs and SNVs we can handle a little bit better than that. So the next question you should raise is like, well, this is great, we can compare tumor normal, but like a lot of cases we don't have normals, right? Or not good tissue was sampled back then. And so we also thought about this. So like when you don't have normal, the pipeline caused this variance, of course, it is a little bit limited, right? I mean, like if there's a normal, this is the best case if it's an adjacent tissue, right? But nevertheless, it can call this variance and give you guys these reports. We have now developed STIX, which is a indexing and population frequency tool for structure variance based on long reads. Up here is the preprint, which really helps you to identify tumor-specific SVs in your sample if there's a normal missing by assigning population frequencies. Since I have a few minutes, so just an example, we used a benchmark data set from NIST, from Genome Model, to assess the SV genotyping power from STIX. And like in Genome, for example, we can assign 30% of the SVs with allele frequency. STIX usually achieves like 85%. So it's really a good bunch of gain here where we can assign population frequencies in different structure variants. And one of my beliefs is that population frequencies is still one of the best tools that we have to identify really cancer-driven or pathogenic variants themselves. There are a couple of explanation marks, of course. And pan-cancer, so Stephen Jones and us work together. They have now also implemented STIX in their pipeline in Canada. So with that, I want to acknowledge a bunch of people. Louis has really been the driver across the different slides. I want to thank the people from the Human Genome Sequencing Center at Baylor College of Medicine, as well as Genentech for a great collaboration here. Some of this work has also been done with funding from NIH, from SMART Consortium, and then just a couple of other funding agencies on the right side. And I want to thank again the organizers for inviting me and you guys for giving me the opportunity to give the talk. Thank you very much.
I want to thank our speakers for two outstanding talks. We are now open for questions, and I think Rashmi is going to read the first online.
While we're waiting, we have one virtual question for Lisa. Although long-read sequencing takes longer for data assessment, is it possible to obtain a limited hot gene list in a shorter time?
Thanks for that question. It's a great one. That's actually how we're approaching the analysis. So the way that we found that this can be most successful in terms of a faster turnaround test is, again, we know that there's a lot of data that are being generated by long-read sequencing. How do we tease apart what's most clinically relevant, what we can approach at this point in time? So we are looking at a gene-based, gene-forward approach, and that's how we're able to speed it along, where it's pretty slick. We take our gene list, compare it to the calls that come out of our pipeline, and we can very easily identify what the partner genes would be within a clinically relevant fusion. Truly is, at this point, a few clicks plus visualization, I'd say analysis in probably under 20 minutes once you have those data in that way. And I think there's still a lot to be gained from that approach, and then it would be starting to build out and look at kind of larger gene lists, for example, but looking for those primary drivers is really effective.
Angela?
Great, thank you. Thank you both for your wonderful talks. I do have a question for Lisa. So Lisa, you mentioned that your limit of detection is about 20%, which equates to about 40% blast, right, if we're thinking about leukemias. Where do you anticipate long-read sequencing fitting into the testing paradigm if you have less than 20% blast? Which is my first question. And then my second question is, where do you see long-read sequencing fitting in terms of testing paradigm at diagnosis, relapse, residual disease, so on and so forth?
Yeah, thank you for those as well. So in terms of overall tumor percentage, you're right, about 40% tumor is approximately what we're seeing. We have dug into a bit how variably are different regions captured in the genome, and unfortunately we're finding that it is variable case to case. So we are taking a coverage analysis into account, especially employing that gene panel. And so then it starts to become a question of which genes are we most interested in targeting for a particular cancer? Can we still find a single or two read support even in a 30x coverage in a lower percent tumor? And we're finding in some cases yes, but in some cases no. So we've also attempted to see if we increase our depth of sequencing coverage, say use 2 SMRT cells, what happens? And that's all active areas of investigation, so it's a little bit difficult to say where we will land, but great question overall. So then with regards to your other question, where does this fit in? Very active area of conversation amongst our group. What our dream would be with unlimited bandwidth and resources would be that a patient walks in, we receive their sample, you prep the sample, put it on a sequencer. Within 2 weeks, you have those data back to the patient with regards to only the structural variant primary driver. A lot more work to be done yet, as I've been saying, probably sound like a broken record on additional changes that we can look at. So that would be the dream. Realistically, we need to fit that into what can our genome core support, what other samples are in the queue, et cetera, et cetera. So all actively being discussed. Thanks.
Thank you. I also have a question for Dr. Lisa Lansdon. 2 questions actually. The first question is, given the limited detection issue, what is your thought of using nanopore sequencing with adaptive sampling to potentially address that issue? Because you can basically enrich on the fly of those regions. The second is, I think long-read sequencing, both PacBio and nanopore can do methylation calling. So have you tried to look into the methylation data, or is there any plan to, in addition to the SV analysis, to classify cases based on methylation? Thanks.
Yeah, thank you for both of those questions as well. So to your first question, we currently are being a little bit selfish, where we want to capture more than just those targeted regions. I think your approach would be fantastic for if we wanted to hone in on the regions that we think are rearranged. It would be beautiful. However, selfishly, we do want to expand, to your second point, to methylation, to SNV, and those are actively being investigated, and with an attempt to see how much can we report back to a patient with, say, a single SMRT cell or 2 SMRT cells, trying to really balance out that cost benefit for the patient. Thank you.
Thank you.
Can I ask?
Go ahead, Jean.
Yeah. Quick question for Fritz. Thanks for tumor lens. Could be very helpful. Are all of these tools, and I'm sorry if I missed it, are they basically platform chemistry agnostic?
Yeah, we mainly tested tumor lengths on nanopore right now, but all of the tools, I have to check them. The methylation requires a little bit of a different tool, quite frankly, but apart from that, all of the tools are both on PacBio and nanopore.
Because I think that's to Lisa's point, different laboratories have available to them different technologies, so making them agnostic to that would be really helpful, I think. And also, Angela already asked, but in your case, you did a very detailed study on purity assessment. What about X? What about coverage for yours? Was that impacting it a lot, the ability for tumor lens to pick them up?
The chromosome X, you mean, or what?
No, sorry, coverage.
Oh, coverage.
Yeah.
Well, we assessed, so the coverages were fluctuating based on the samples a little bit, right? But all the samples that we tested so far were at least one flow cell. And so that gets us, sniffles can go down to 6%, 7% variant allele fraction with that. And the other work from NIH that we are currently doing from the SMART consortium is going actually even below that range. It's the Somatic and Mosaic Consortium in Human Tissues. But there you need more coverage, right? So as soon as you go below 5% variant allele fraction, you probably need 2 flow cells or more, depending on how low you want to go. But yeah, it's a very good point. All the tools are available and work on PacBio as well as nanopore. The methylation caller, there's a different caller for that methylation part that needs to be swapped, but there's a tool available.
Thank you.
Okay, so I'd like to thank the speakers again for a wonderful session, and I'm going to leave Rashmi to introduce our keynote.